Thank you for joining us. We're going to hear today about Below the Seafloor, How Microbes Survive and Thrive in Extreme Environments by Melody Lindsay, and I'm going to introduce her in just a minute. But I want to thank our sponsor. HM Payson is now Corient, so thank you HM Payson, and thank you the new Corient. They have been long-term sponsors of this event and have a long history with us, so we are very, very grateful. Back to Melody. So Melody Lindsay came to us a number of years ago as a postdoctoral scholar with Beth Orcutt, who is our Vice President of Research. She was just an exceptional scientist, so she applied and was promoted to a research scientist. And then last year, we did a national and international search. We had hoped at the time to hire 4 to 6 new people, new scientists, each of which would come in and build their own research group. When the federal pause in funding happened, we decided we should probably not bring in new people and make sure that we could take care of all the people that we already had. But we asked the scientists if they still wanted to make offers for a new senior research scientist position, and it was unanimous, and one of those people was Melody Lindsay. So I think she really kind of blew everybody away because, one, she is scrappy in terms of thinking about this new world and the new funding environment we're all finding ourselves in, and finding new ways that she can use her tools to address the questions that she wants.
Thank you so much, Debbie, and thank you all for being here. I'm thrilled and honored to be here today to talk to you about some of our ongoing research and also to take you on a little trip below the seafloor, investigating how microbes survive and thrive in some of the most extreme environments on Earth. And a bit of a spoiler alert, survive is more a better description of what the microbes are doing in these deep ecosystems. Thriving, maybe not so much, but they're managing to live, so that's important. But before we go below the seafloor, I'm going to take you to outer space for reasons that will hopefully be clear after the break. So why do we search for life beneath the seafloor? My role as a geomicrobiologist, as a scientist, a lot of my science has applications to astrobiology and looking for life in environments on Earth that might mimic environments that are found elsewhere. With this work, we're developing some really cool methods to detect life in these extreme ecosystems. These methods are of great interest to NASA, which Debbie mentioned as that award, so fingers crossed. But then we're going to have a slight break for questions, and then I'm going to take you on some recent scientific ocean drilling expeditions that I have been a part of, and we're going to end by investigating our own backyard, the Gulf of Maine coastal subseafloor. How are you all enjoying living or visiting Maine? Do you really like it? I'm a big fan of Maine, which is why one of the reasons why I fought so hard to stay here, but also Bigelow is a wonderful place to work and to get science done. I really love living in Maine. Do you maybe go for a swim this week or last week, maybe when the weather was a little better? No? Okay, not yet. Maybe in August in a couple days. So I really like living on planet Earth in general. It is the only place where life is able to exist that we know of so far. The habitable zone, one of which examples is this lovely mid-coast Maine environment, is comprised of a bunch of different parameters that come together to form just the right ecosystem for life to live in, including humans. So this is a wheel depicting different aspects of what makes an environment habitable. These include things that are important, such as temperature. I personally like it at a balmy 68 degrees Fahrenheit, like all the time. Winter's cool too, though. Pressure is also very important. We exist at a pressure that allows us to exist as life forms. We have the right amount of salt. We have the right amount of nutrients, and also not too much radiation, but just a little bit. Another very important aspect of a habitable zone is having sources of energy or sources of reducing or oxidizing power in the form of different chemicals, electron donors, electron acceptors. So Earth, our lovely home, this is a photo taken from the Artemis II mission, which I was just so inspired by earlier this year. It was just the coolest thing. But this beautiful photo of Earth really makes me think of how lucky we are to be on this habitable planet. We have a nice atmosphere, great oceans, but also a lot of land to live on. This diagram down here that I'm going to show you a series of depicts 4 different aspects of habitability that are really important to both life in general, but also humans. So pressure, pH, temperature, and salinity, all really important for life. Since we live on Earth, we get to experience a wide range of these. Humans like it at a range very narrow range of pressure, pH, temperature, and salinity. So this is what kind of the human habitable zone looks like on Earth. But if you were a microbe, specifically a microbe like a bacteria or archaea, some of the single-celled forms of life that we have here on Earth, you might like it elsewhere. You might live in a boiling hot spring, which is pH 1. I don't think I would enjoy it very much at pH 1. That's like battery acid. You might live at 40% salt. I happen to like living at either 0% salt or the ocean, 3-ish%. So if you're a microbe, you might be able to live elsewhere on Earth that humans are not able to live in. When we look at the habitability zones of other, or the ecosystem parameters on other planets, such as NASA, or other moons, such as Europa, which is another ocean world, those zones which are present on these different extant bodies change a lot. There's a different range of pH, pressure, temperature, and salinity that are possible there. And so maybe even though it's not habitable for humans, it might be habitable for a microbe that is able to live in an extreme environment on Earth. So I've kind of introduced the concept of habitability, and I've talked a little bit about microbes. I was getting ahead of myself. But when I talk about microbes living in extreme environments, in particular, I mean bacteria and archaea. So this is a tree of life showing the 3 domains of life. We have bacteria, which is this massive spread of different lineages of life on Earth. We have the branches for archaea, and then the eukaryotes, which on this tree are a little bit of an afterthought. If you're wondering where you are on this tree of life, you are somewhere in this mix here. But these microbes are incredibly diverse in terms of their genetic content, what they encode for in their DNA, their identity. They're also incredibly diverse in terms of metabolisms, which I'll get to on the next slide. One thing they're not as diverse as eukaryotes are is in their shape and size, their physiology. So this is, I don't want to say this is as good as it gets because some people in this room have really great microbes that they've been studying, really beautiful physiologies, but you generally are stuck with a circle or a rod and different manifestations therein. But they can do really cool metabolisms. That's where the beauty, I think, a lot of the beauty of microorganisms are. So especially in extreme environments, microbes can subsist and exist based on using different metabolisms, wildly different metabolisms. So us as humans, we conduct aerobic metabolisms, basically breathing. We use oxygen. In particular, we use things like glucose, which is a carbon-based compound, with oxygen, and with that respiration metabolism, we produce CO2 by breathing, as well as water, and then we also get energy from this process. If you were instead an anaerobic microbe instead of an aerobic organism, microbes can also be aerobic, an anaerobic microbe is able to do so many other things than utilize oxygen. One such process that I'll focus on for the latter half of this talk is the process of sulfate reduction. And so instead of breathing oxygen and using a carbon compound such as glucose, a sulfate-reducing organism can breathe sulfate, use organic carbon, and produces other compounds such as hydrogen sulfide and bicarbonate. This is not the only anaerobic metabolism out there. Microbes can also metabolize hydrogen, manganese, iron, so many more that I didn't fit on this slide here. The metabolic processes are just so diverse and incredible. So I'm a geomicrobiologist, but I also consider myself an astrobiologist because a lot of my geomicrobiology research has applications to astrobiology, looking for habitable worlds and looking for if life could exist elsewhere, potentially. So we must first understand life on Earth to have a chance at finding life elsewhere. Our current total examples of life in the universe that we currently know of is n equals 1. There's one example, and that's our lovely planet Earth. So the question becomes, what environments are most similar to extraterrestrial worlds, such as Mars or Europa, that we can explore and understand here on our home planet? This is a diagram showing a wide variety of different environments that Earth hosts. I don't expect you to read all of them, and I'm going to focus in on some of these. For my PhD work and work before my PhD, I was very lucky to be involved in several different projects in a variety of extreme environments, and I will tell you briefly about some work in hot springs and hypersaline lakes. Hot springs in Yellowstone was the focus of my PhD work at Montana State. This is me standing in front of this belching sulfur pit somewhere in the backcountry of Yellowstone, hanging out with the bison and the bears, if you've ever been to Yellowstone. But Yellowstone is home to this incredible array of extreme environments. And so this hot spring was about 80 degrees Celsius, pH of 2, really low pH, battery acid again, boiling water. But in these hot springs, my work demonstrated that the microbes that are living in these hot pools do eat hydrogen that is produced from water-rock interactions in the subsurface. So these windows in the subsurface are a great example of how geology impacts the chemistry of the system, which then fuels the microbiology or the life that's able to persist. On the flip side, I also had several projects that weren't my thesis, but I got distracted easily, come out of the Great Salt Lake. So this is the north arm of the Great Salt Lake, which is about 38% salinity. We can compare that to ocean water, which is an order of magnitude less in salt content. And so in this system, these microbes, the capable of photosynthesis as well as sulfate reduction, that anaerobic metabolism, construct these microbial-like rocks. They manifest as these little salty pillows you can see here, and I liked this photo because it mimicked the clouds in the sky. So this is a good example of how biology impacts chemistry, impacts geology. But today I'm going to focus in, after taking you on a journey around the world and elsewhere, on deep sea sediments as well as the marine subsurface. And so I'm really interested in the subsurface because of how important the subsurface is for life on Earth. Subsurface is the largest environment that we have on Earth for life. You're currently standing on top of the subsurface. No matter where you go, even if you're flying in an airplane, there's still the subsurface beneath you. There's a lot of subsurface. Specifically, there's a lot of marine subsurface because the ocean covers 70% of the planet. So in the subsurface, this is a plot showing the distribution of different forms of life on Earth. So plants, fungi, protists, animals, they live in generally a mixture of the terrestrial and marine surface environments. But when we get to bacteria and archaea, which are 2 out of the 3 domains of life, so they're very important, they comprise 80 to 90% of the biomass. The 80 to 90% of bacterial and archaeal biomass lives in the subsurface. These subsurface environments generally don't have influx of energy or nutrients or chemicals from photosynthesis, which is a really important surface process, but instead they rely on anaerobic metabolisms, which use chemicals found in the subsurface. So despite the vastness of the subsurface and how, pervasive it is on this planet, these extreme environments can be really difficult to access. It's hard to dig deep enough to get at most of the subsurface. And they're also really low in biomass. So even though they're very vast, everything is in low concentrations because rocks get in the way of life sometimes, but they also aid in life. So they're low in biomass, they're low in nutrients and chemistry, and also in energy. However, these subsurface environments and these particular aspects of low in biomass, low in nutrients, and low in energy might be similar to environments found elsewhere. So I mentioned before, rocks kind of get in the way of life, but the subsurface has a lot of features. It's not just one solid mass down there. There's a lot of things that are conducive to life living in the subsurface. This is an artistic depiction of a subsurface lake on Mars. I think they've been able to detect that there might be these subsurface features that are filled with liquid water that might host life on Mars. Currently, it's not accessible by this friendly face here, the Mars 2020 Perseverance rover, but maybe one day we'll be able to access subsurface environments on Mars. In the meantime, we can certainly access subsurface environments on Earth. I first got my start in geomicrobiology and science as an undergrad because of a project that I was involved in that brought me to gold and diamond mines in South Africa. So this was my first field experience. This is a map showing where we were in the gold mines of South Africa, a little bit south of Johannesburg. This is a plot it's a 3D image of what the subsurface features look like in a gold mine. So this particular gold mine, Katsong, you have a shaft dropping about 3+ kilometers, 3.1 kilometers into the earth. So as an undergrad in 2012, here's a picture of me on my first fieldwork experience in the mine. But I did not quite realize what I was getting into, but I got into an elevator, which they called a cage. And you are crammed in there with all of the mine workers who were very nice. I'm sorry, I'm bringing, but then you drop 3 kilometers in one single elevator shaft at a speed of about 60 kilometers an hour. I'm sorry, I don't know what that is in miles. But you drop incredibly fast. You can feel, and you're completely in the dark when this happens because they don't bother hooking the cage up to electricity. So you're dropping, you can feel the heat from geothermal heat because you're dropping so far into the earth crust. You can feel the heat rising. You can smell kind of the sulfuric fumes that come out of the earth. And it really feels like you're in a Jules Verne book, Journey to the Center of the Earth. So go see that movie. I love that book. Or yeah. Funnily enough, there's a microbe that they isolated out of the gold mines in South Africa that is named after hell. So anyway, but I honestly thought it was a pleasant experience. It's very humid down there. There's a lot of water. And so it kind of feels like you're at the spa. But so it's very warm and, yeah, anyway, and we were there for the water. And so this depiction shows all of the mining activity up here and then some drilling that they did into the side of the mined walls, which actually accessed aquifers that are 3 kilometers beneath the surface of the earth. They host water up to 2 billion years old. So that water has not been touched or perturbed for 2 billion years. And we were able to find active microbes that lived in there. And so that was I was lucky to be involved in that as an undergrad, as a paper published in 2016. But to more recent endeavors, now I'm going to tell you about some of the new technologies and methods that we are leveraging here at Bigelow to determine microbial identity as well as microbial activities in different environments. So I'm going to take you on a quest to several different environments to detect and measure microbial activity in diverse environments and to link together microbial identity and activity. And so when I say identity, what I really mean is the genome of a microbe, which is what is encoded in their DNA. We now have the technology, thanks to folks at Bigelow and technology we have here at Bigelow, to link that what's encoded in an organism's DNA to the function that they're actually actively doing in an environment at any given time. We can do that at the level of a single cell. So we have thus far done this in the surface ocean, in the deep continental crust. And what I'm going to tell you about today are just some brief examples in the oceanic crust, coastal marine sediment, as well as some processes that we were able to look at in deep marine sediment. So focusing on the subseafloor ecosystems for the latter half of this talk. So before I break for questions, just to direct us to where we're going to go in the second part of this, we're going to investigate how microbes live in deep subseafloor sediment and rock. Sediment, especially marine sediment, is really important for life. Again, going back to the idea that the ocean covers 70% of the planet, and there is something that lives underneath the ocean in this really thick sediment, sometimes maybe not so thick sediment, but also the rock underlying that. These environments are really important for microbes and life in general, because life can live there even though there's solid stuff in the way. And really it can capitalize on these solid pieces of rock or sediment. Microbes can live in the planktonic pore spaces where water is able to flow. They can burrow into different sediment or rock particles and really make a good living for themselves there. And they're just, living off of the chemical forms of energy that they can get out of these water-rock interactions. But before we go there, I'd like to invite any questions.
Your last screen, you were showing very thick concentrations of sediment. Is that because it's flowing off the
Yeah, exactly.
continents
Yeah,
into the
it's
soil?
the runoff. It's a lot of this is the runoff from river input. Yeah, from the continents. Yeah. Especially here and here.
I'll ask a question. So you're not claustrophobic?
No.
Good.
Yeah, no. I have other faults when it comes to field work.
I hope claustrophobia is not a fault.
Yeah.
I think it's a sign of sophistication.
How about seasickness? No, I'm kidding. At the time, I didn't really realize what was happening. I think that helped a lot. Yeah.
I assume there are others around the globe doing it. Do you get the opportunity to interact
Mhm.
and sort of direct all your work together so you can sort of get to a common goal quicker or result quicker?
Yes. Yeah, that's a great point. So actually, and I'll go back to this example of the work in the South African mines. I actually was just on a Zoom call today because we're going back to this mine to deploy some of these novel technologies. But I was struck, it was a Zoom call with drillers in South Africa, with seismologists in Japan, and some microbiologists in the USA. And we really, we would not be able to do this work without them. And I feel like, especially in geomicrobiology, it's a very interdisciplinary field. And we often have shared goals with the same samples or the same sampling regime. And so even though you'll see this when we take a trip on the drilling vessel that I'll tell you about in the second half, but it's really, we get the same samples and go to the same place with different goals, but they all dovetail together to really understand the system in an interdisciplinary way. Yeah, that's a good question.
Oh, I was wondering, is the thought that the organisms that live in these more extreme environments are organisms that originated on the surface and then are exploiting these areas, or are these, or is this where life originated and then came up to the surface, when oxygen became more available?
Yeah, that's a great question. And so there are many hypotheses for where life originated, and one of my favorites, I guess I'll go with favorites. One that I think has the most support is the fact that life on Earth may have originated in a hot subsurface environment. When you look at a tree of life, which I will go back to here, this is color coded according to the identities of the microbes. But actually, if you looked at the temperature that each of these different branches, each of the representatives that are represented by these branches can grow at, the thermophiles, the ones that grow at really high temperatures, are the more deeply branching ones. And so those are that's kind of like the ancestors of all the rest of them. So the most deeply branching ones are thermophiles. So we think life might have been the last universal common ancestor, the progenitor of life, might have been a thermophile, and it might have been living underground because they don't really tolerate UV radiation that well.
I have a question about how you obtain your samples and eliminate the possibility of contamination from outside.
That's a great question. It's a very important question. I've spent a lot of time trying to eliminate contamination. And I think my philosophy now is we do the best we can on eliminating contamination. For example, in the second half that I'll tell you about, we take large samples, but then cut them down so that only the pristine middle is what we actually sample. But we're humans and we exude a lot of human microbes and human cells. So we're handling these things in a not so sterile environment. So we have a really good sense of what microbes do represent contamination versus what we expect to find there. Sometimes that doesn't always work out perfectly, but yeah, we do our best to eliminate the contamination, but then we also try to expect contamination and know who that is. We also run a lot of blanks, so testing for contamination directly.
Is there any chance that these subsurface microbes, should there be a huge catastrophe, such as huge nuclear war or something on the surface, is there any chance that they could keep human life going?
That's a good question. So actually, I've thought about that, not an insignificant amount, because so another finding from the work in the deep gold mines of South Africa, number one, these microbes actually subsist based on the reactions of radioactive chemicals with water. And so that radiolysis of water, thanks to the uranium in this gold mine, is actually what fuels the microbes. And there's something else that lives down there with the microbes, and it's called Mephisto. That's the hell reference. But it's this nematode worm that eats the microbes, that eats the uranium products. So if a worm can survive down there, maybe humans could find a way to harness that and survive. So for this latter half, where we're going to investigate how microbes live in subseafloor rock and sediment, I'm going to take you on 3 trips, the first of which is a scientific ocean drilling expedition on the drilling vessel Joides Resolution. So this is the Joides Resolution. She is a repurposed oil exploration vessel. This photo was taken maybe a decade ago, where it was sailing out of a port on this, if you know where this is the island of Oahu in Hawaii, and I couldn't resist showing this picture. First of all, it's a really nice picture of this drilling vessel, but also I lived here for most of my childhood, so I like to think of me growing up in Hawaii while the Joides was sailing by a couple decades ago, maybe. But to introduce you to the drilling vessel Joides Resolution, it's a 470-foot long ship. It is for scientific ocean drilling, again, repurposed from oil exploration. But all of the scientists' labs and living quarters are restricted to this front half of the ship. The rest of the ship is dedicated to drilling, which is the whole reason why we're on the ship in the first place, so it makes sense. We have this drilling derrick here, where they are able to drop the drilling apparatus, and a lot of the ship is dedicated to storing and accessing the kilometers, these miles and miles of drill string. And so and by string, I mean actually a very thick metal pipe that they use for drilling, because they have to reach through the ocean, through sediment, oftentimes to hard rock, and that is several tens of kilometers of drilling that they have to get through. So being on the Joides Resolution and drilling for core is just an immense effort, and I have to thank the crew, staff, drillers, technicians, and scientists of the expedition that I was lucky to go on in the summer of 2023. IODP stands for International Ocean Discovery Program, or a couple years ago it was called the Drilling Program, and we were on Expedition 395. So this is all the folks involved there. And so, yeah, they're just an incredible group of humans who are really supporting the science and the samples that we were getting on this expedition. So what is a sediment core or a rock core? What is a core? So I thought it was funny that, when I think of a core, there's 2 definitions. The not scientific definition is the core is the most important part of an idea, group, or object. And it was definitely the most important part of getting core on a scientific ocean drilling expedition. But in our case, it means long cylindrical samples of mud, sediment, or rock. Not ice, but you can also have ice cores. I brought props. So this is a piece of a sediment core. See this lovely mud here. So in this case, it was mud, but we can also have lithified sediment, which you see here. We split that open on the ship to reveal these beautiful layers of deposited sediment. And so just incredible colors. I don't want to misspeak, but I think there's some glauconite in there in case you really like rocks and minerals. Please do not ask me any questions about this. but I know the microbes that might live in there. So these beautiful layers of sediment, we also sampled for hard rock. But we were drilling for core in the North Atlantic. So we were at a location off of the Reykjanes Ridge, which is the northern part of the Mid-Atlantic Ridge, a little bit south of Iceland. So the Reykjanes Ridge in this schematic, I'm indicating the Reykjanes Ridge in this schematic of what the Earth would look like if you took a cross-section through the bottom of it. So this part of the schematic here is actually water, even though it's white. Then we have the sediment layers and their depth lying on top of hard rock, which is again represented in white. But at the Reykjanes Ridge, we sampled in a transect out towards the ridge flank at these 6 different sites, so 5 sites along the ridge, and also a sediment site off of the coast of Greenland, which was really fun to visit as well. This is maybe a more familiar depiction of where we were in the North Atlantic. Here is Iceland up here, the western coast of Iceland. If you've ever looked at a map on Google Earth or flown over the Mid-Atlantic Ridge, in this particular region of the Reykjanes Ridge is home to these V-shaped troughs. And we were there along with a bunch of geologists, sedimentologists, other scientists who were not microbiologists to investigate various ideas of what was causing that to form. But it was really exciting being a little bit south of Iceland. One particular notable thing that doesn't happen every drilling expedition is that we were fortunate to get week 6 deliveries straight from the Icelandic Coast Guard. So I just have a short video to play. I like to show this for two reasons. It gives you an idea of what the North Atlantic seas are like. It's a little bit rough on this particular day. But yeah, the Icelandic Coast Guard came to visit us. They brought us dynamite, which was a safety thing. We had to have dynamite in order to safely drill to the deep depths of basalt that we wanted to extract from these cores. And they also brought us lettuce because it was week 6 and we had run out of vegetables in week 4. So that was very exciting. Oh, and bananas. Somebody really wanted bananas. Yeah, anyway, so welcome to the North Atlantic and the Icelandic Coast Guard ship Thor. So it wasn't always like that. I have never seen the ocean as different as it was in this North Atlantic expedition. Here is the microbiology and chemistry team taking a brief break, not too long because we were an American-run ship and we had to get back to work. But I had never seen the water so serene and calm, and so it was just an absolute beautiful sight to see it so calm. But also it was cool to see, you know, kind of the rough North Atlantic weather happening. We had some wildlife come visit. They loved checking out the drilling. There's a dolphin that I didn't get to see because I was probably working in the lab. We got to see other life as well. I just want to show you a kind of humorous video. This shows the drill string, and so a string is a bit of a misnomer, but a drill string, which is this giant metal pipe going down through the water column to the bottom of the ocean. And we had another visitor come because they were curious about the drilling. And so you can see, the squid hitting the drill string. I guess it either really wanted to get core or it was angry that we were there. But yeah, he swims off, so hopefully he's fine. Okay, so we were on this drilling ship in the middle of the North Atlantic. It could be pretty rough, but most of the time, honestly, it was pretty calm. But it was really nice being on this ship. I actually get really, here's a bit of a side. I get pretty seasick, but this ship had stabilizers, which was great. So even though we had, 5-meter swells at times, the ship would drop these stabilizers. And so in a drilling platform, say in the Gulf of Mexico, these legs might come down and you're anchored to the bottom. We couldn't do that here because we were 2 kilometers from the bottom of the ocean. So instead we have the drilling vessel has these fans on stabilizers and they drop down all over the ship and they make it so that the drilling can happen and it's not so wild of a ride. So that's a sped up video of that stabilizer dropping. I also wanted to show you some drilling in action. You can see on this drilling derrick here, the pipes are going up and down, and that's actually the motion of getting the core out as it's being drilled. I just want you to note the timestamp of 1 in the morning, hard at work, 1 in the morning, American-run ship. Anyway, yeah, I said that for you, Debbie. Anyway, so these cores come up in 10-meter long sections. I have this for scale. I'm not going to move too much. But this is less than half of the different core sections that would come up every hour. So we had a really fast rate of coring because the sediments were nice and smooth. So we were able to get about 10 meters per hour. And so when the cores came up, they would come up from such a deep depth that they were highly pressurized. And I just wanted to share this video with you. This is a core laying on its side. It's a 10-meter long core. And we would have to drill holes into it to release the pressure once it came up because it's coming up from 1 kilometer deep in the sediment, but then there's also 2 kilometers of water stacked on top of that. So really highly pressurized system. So the sediment is just kind of oozing out of the core there. So I was there alongside all of the geologists and paleoclimatologists and chemists and petrologists, all wonderful humans doing wonderful science. I was there to ask the question, what do microbes do to survive in deep sediment? So I'm going to show you how we process samples in order to answer that question. So this is our 10-meter core and all of the text carrying the core as it gets taken out of the derrick drilling system. Every time a core came up, I would grab a 5 to 10 centimeter long piece. And sometimes it looked like that. And you may be thinking, that looks like a rock. That doesn't look like mud. It's highly lithified mud, again, from being squished at the bottom of the ocean for so long, millions of years. We handle this in a relatively sterile environment. We have these fans that create this vacuum that doesn't allow our human microbes to get on to the core. I have to shout out Justin Dodd here for sampling all of the microbiology on the off shift so that I could actually get some sleep at some times. So he's also a great chemist. So we not only sampled sediment, we also sampled hard rock. And so it looks a little bit different than the sediment. Hopefully you can see that. You can see some of the white, which are these lovely veins of calcite going through the basalt iron-rich rock here. Every time we sampled hard rock, the petrologists were really interested in their own research questions, but as a microbiologist, I wanted to know who was living there, and I had to sample it relatively quickly so as not to introduce contamination. I also, even though we had been quarantining at this point for 6 weeks because we were alone on the ship, minus the lettuce that came over from the Icelandic Coast Guard, I still asked everyone to wear masks so that we weren't breathing onto these pieces of core that housed deep subsurface microbes that are low in number. So once we got these pieces of core, big thanks to the research techs for facilitating this, I would put it into this stainless steel box called a rock box. We would light it on fire. It's a little hard to see there in order to sterilize the environment for the processing. And I was so surprised at the difference between how hard basalt rock is from heavily lithified sediment. Lithified sediment is nothing compared to cracking open a piece of basalt. So this is me chasing a piece of basalt around this sterile box with a sterilized hammer and chisel, trying not to make it explode out of the box from hitting it. So fun sampling. And I'm wearing hearing protection because it was really loud hitting the rock around. So once we have our piece of core, we use a series of dilution, sonification, and centrifugation steps to separate out the cells from the sediment or rock particles. And so that's just an artistic depiction here. So we're just trying to get cells out because we want to put these cells through a machine called a flow cytometer. A flow cytometer is a machine that works with an aqueous, with fluids. It doesn't really work with rocks. You can't take a rock and put it through a flow cytometer because it uses fluid to line cells up one by one in order to analyze cells for fluorescence, for size, for other cell aspects. So a flow cytometer is a machine that lines up these cells one by one. We can get a lot of information out of it, but it also serves as the initial step in sorting cells for single cell genomics. And so these are the technologies that Bigelow has really developed and has pioneered in terms of this low biomass work that we're working on. So single cell genomics takes a single cell that comes out of a flow cytometer, these cells that are lined up. We can isolate single microbial cells, one cell per well of this 384 well plate. So a cell sorter, the flow cytometer, is able to deposit one singular cell into each one of these wells. Within each of one of these wells, we can add reagents to amplify the DNA and also sequence the genome of the microbe that is in this one single cell. So with these methods, flow cytometry and single cell genomics, we can get cells from the subseafloor, from really deep sediment, even though the cells are low in number and present in these lithified sediments. So I'm going to show you some data of our cells that we were able to capture from the subseafloor from sediment that represents a break in 3 million years. And so these are cells where each dot represents one single cell that we know the identity of, and we also know what the function is encoded in their genome. These cells are from 17 meters below seafloor. 17 meters is honestly a pretty good distance when you're talking about the subseafloor. It's hard to drill 17 meters down. That's deeper than a lot of the wells, I think, that people get drinking water from. But we went further, and we were able to get cells from 232 meters below seafloor. So this is the first instance of single cells that we were able to capture, single cell genomics from 232 meters below seafloor. When we look at how long it took for sedimentation to go from 232 meters up to 17 meters as sediment accumulates, that represents a time frame of 3 million years between when the sediment at 232 meters was deposited and the sediment at 17 meters was deposited. So just an incredible amount of time represented in this sediment. One thing I want to note from these flow cytograms, that's what these plots are depicting. The axes are not so important, but the distribution of the colors, which indicate identity, is important. And what really struck us when we first looked at this is how remarkably similar these colors are. So there's the same community we see at 17 meters and 232 meters. Digging a little deeper into the genomes of these cells, in these red boxes, we have a lineage called Aerophobota. That's a microbe that's capable of fermentation. And so we see the same exact species of Aerophobota present at 17 meters that we do at 232, so 2 meters. So they're really persisting throughout this deposition process and across immense timescales, 3 million years ago, 3 million years. Do you imagine living next to the same neighbor for 3 million years? That's a long time. So overall, these cells are capable of fermentation, and thanks to work by Senior Research Associate here at Bigelow, Tim D'Angelo, we have found that these cells select for genes involved in cell wall maintenance and repair. So they're just taking care of themselves in order to persist or to reproduce over these long time scales. And again, this represents single cell life recovered from sediment over 3 million years old, which is incredibly old. I'll leave this part of the talk on a bit of a funny note. This cartoon was published while we were at sea, so it was published in 2023. I don't know how many of you recognize this cartoon. I love it because it's all like ocean animal based. But this was a cartoon about ocean drilling, which was really exciting. You can see the drill string or the drill pipe here. And basically, what this cartoon was depicting in 2023 is exactly what we've kind of seen with our excellent data. So there are things living under the bottom of the ocean. Yes, there are things living in there. A big community of microorganisms. But yeah, they hang out in the mud and they don't do a lot. They're kind of eking out a living, doing fermentation, which is one of the most basic microbial metabolisms. And yes, I would have been good at that. I would like to live in the mud. Kind of do sometimes. So maybe not so much surviving, maybe a little bit more thriving. I'm going to take you on a brief journey from no longer in the North Atlantic, but now to the Juan de Fuca Ridge off the coast of British Columbia, where we're looking for active microbial life in the Juan de Fuca oceanic crust. So at this location off the coast of British Columbia, this is the site of several of these devices called corks. And this is an artistic depiction of it, my artistic depiction, I apologize. But these are well devices that plunge through the sediment into and access fluids that cycle beneath the sediment. So through the basaltic hard rock at the Juan de Fuca Ridge. So fluid flow here, we hypothesize, is supporting active microbial processes. So these corks, these devices called corks, were installed by drilling vessel JOIDES Resolution, but we were able to access them with the research vessels Atlantis and Thompson using the remotely operated vehicle Jason. These are cruises led by Dr. Beth Orcutt. I'm just going to show you what accessing the cork was like. So here is a video of the remotely operated vehicle robot Jason coming up to the cork. I have, in contrast to this video showing all of the growth and deposition of life on this cork, what they looked like before they were installed at the bottom of the ocean. So some buildup, some nice graffiti. That was not done by things living on the bottom of the ocean. But I thought this was funny. Before we accessed some of the ports, the ROV Jason had to clean some of them off with a toilet brush that we got from the local Oregon Fred Meyer. So enjoy that photo of a toilet brush. I'm sorry, I don't have the video. Now getting to sampling the cork, the ROV Jason is able to manipulate what we call squeezer syringes. And so these are syringes, it's at the top of the cork. The ROV will squeeze it and you can see the water being collected in the syringe. You'll notice that there's this kind of shimmering effect. It is not only because my video is a little delayed, but it is mostly due to the mixing of 65°C crustal fluids coming out of the top of the cork, mixing with really cold bottom seawater. And so that's why you have that thermal difference and you can see that shimmering. So within this crustal fluid, we found organisms that were indeed active, which was a huge, huge finding, I think. We're still working on publishing this, but we have these communities of sulfate reducers that are capable of producing sulfide from this anaerobic metabolism. Each one of these dots represents a cell that we were able to sort and sequence with flow cytometry and single-cell genomics, respectively, and we see a wide spread of activity that these organisms are doing. So overall, at Juan de Fuca, we have active cells metabolizing sulfate. A femtomole, which you'll notice on this graph here, femtomole is 10 to the minus 15th mole. So that's very, very tiny amount. We can compare that to a human like you or me who respires 22 moles of oxygen per day. Now, a human and a microbe are not exactly comparable, but if we compare the rates that we were able to capture at the Juan de Fuca marine crustal subsurface with rates that we have previously published in the terrestrial deep subsurface in an aquifer beneath Death Valley, these are still orders of magnitude lower. So they're really slow, more so in the terrestrial deep subsurface. It lives up to the name, slow life in the fast life. So I'm going to end this part of the talk on a little bit of a depressing note. This is a map showing where we were able to sample with the drilling vessel JOIDES Resolution and the US Scientific Ocean Drilling Program. This bottom map in black shows the regions that we are no longer able to access because the JOIDES Resolution has retired. We no longer have access to most of the world's ocean subseafloor, despite scientific ocean drilling being mandated as a priority by Congress. With the retirement of this US drilling vessel, there is no plan for the US to have ocean drilling anymore, except for some mission-specific platforms by the new Office of Ocean Drilling. There are also other vessels internationally that are capable of going into deeper waters. There is a drilling vessel that Japan uses, but it doesn't leave Japanese waters. But China also has 2 new drilling vessels coming online. So maybe internationally, we will still be able to go to these places in the near future. In the meantime, until those drilling vessels maybe become more available, there's still lots to discover in places we can still access. I love this quote that I was recently introduced to, but in the mud and scum of things, there are always something sings by Ralph Waldo Emerson, and makes me think of a project that another Emerson and I have been working on, Dr. Dave Emerson, a scientist at Bigelow. He's depicted here, and we have an ongoing project in Maine coastal sediments. These coastal sediments are easy to access, relatively understudied, but are high in biomass, so not low in biomass like the ultra deep subsurface. We've involved a lot of students, and I want to shout out one here, Eliza Goodell, who did incredible work on this project that we published in 2025. In this environment, there's a lot of different biological things happening in the system. The tidal influx of seawater contains fresh nutrients and carbon sources that the microbes are able to capitalize on. And there's a lot of bioturbation. In this photo, you can kind of see some clams. There's often worms burrowing in that bring oxygen into this oxygen-poor sediment. Flies like to burrow into, and then humans, either through aquaculture reasons or through collecting sediment. So there's a lot of cool stuff going on here, but we are funded by NSF to do current work, bringing this a little further afield. This work, and actually this sediment core, was taken from the Edgecombe Eddy. I don't know how many of you people drive by the Edgecombe Eddy if you go through Wiscasset, but if you've ever driven by there and have smelled a wonderful rotten egg smell, especially at low tide, that is thanks to the microbes, and specifically the sulfate reducers that I've been talking about. So lots of cool things at the Eddy, but we are now funded to take the Bigelow research vessel Bowditch out to do some Maine coastal sediment out towards, more towards the ocean, not in the tidal flat. So I'm just going to take you on a little journey on a sediment core. So imagine you're a sediment core. Actually, it's this sediment core right here. So yeah, you'll see a sampling sediment at the bottom of Harpsley Sound. I don't know if you could see the cloud of sediment, but you can see it kind of blowing off of the sediment core here. We bring it back on the boat in order to process it to look for the microbes that are living in coastal Maine sediments. And so this core here, we're able to capture a sediment core about 1.5 meters long, And that represents several thousand years of sediment deposition, can represent up to several thousand years. So still really cool things going on here. So once a sediment core is back on the boat, this is from a few days ago, actually, and thanks to Ann Madden for this photo and video. We have Dave, Anna, and Ariel here all frantically sampling the sediment core once it's back on the boat. We sample for chemistry, we sample for microbiology. I just wanted to share this quick video of us getting the sediment core back on the boat. And we have a friend make a brief appearance, which we'll hopefully see in a second. But yeah, we're hyper-focused on getting the sediment safely back, but then the whale comes. The whale really wants to know what you're doing with that core. Why is there this thing dangling from the boat? It really, truly, the whale just wants to know the life histories of sediment-hosted Piteski bacteria and Chloroflexota, which is what we're after too. So yeah, big thanks to the whale for making an appearance. Thrilling to see wildlife, but yeah, we were focused on getting the sediment. So I want you to take away from this talk that life is found in unexpected places. The subsurface is home to the vast majority of life forms on Earth. We now have the technology to interrogate it at a level never before resolved. And this life on Earth tells us a lot about the ways microbes might be able to live, emerge, and evolve here and elsewhere. So a big thanks to those involved in all of this different research. Could not do it without all of you. Absolutely wouldn't be here without all of these people. But a big thanks to Beth Orcutt and Dave Emerson here at Bigelow for hiring me as a postdoc in 2019. A big thanks to Ramunis and Nicole as well for all the work that they're doing in the Center for Aquatic Cytometry and the Single Cell Genomics Center. Thanks to all of the folks involved in the different cruises and different field work that I've told you about today. And thank you to our funders, NASA, Exobiology, and also NSF. So thank you. Thanks for going on this journey to outer space and also bottom of the ocean.
Thank you. Could you go back to the slide showing the prohibited ocean areas?
Oh, yeah.
Why are you not allowed to drill wherever you want to drill?
Oh, yeah, it's not a matter of being allowed. And in fact, these open ocean areas are often the ones that are outside the exclusive economic zones of different countries. And it's not that we're not allowed to, it's simply that we can't with the technology that we have available. We cannot reach down there.
Would you or could you get different information if your sediment cores were scanned as opposed to dissected?
That's a great question. And actually, as a part of the workflow for scientific ocean drilling, I'm trying to see, most of this core, aside from what we take for chemistry and microbiology, kind of these ephemeral measurements, they do stay intact and they get scanned with X-rays and gamma radiation. And it can tell us a lot about like how much iceberg rafted debris are in these. What is the sediment porosity? What does the sediment look like? Unfortunately, we do not have a way to do that with microbiology or the aqueous pore fluid chemistry, but that would be really cool if someone did that. But yeah, I have colleagues who were on this ship who did do X-ray scans and gamma scans. Yeah.
Melody, you mentioned a few times that you have like thousands of years of sediment deposition or 3 million years between parts of the core. Like, how connected are those communities, or do you know that? And yeah, how connected are they? Can they migrate?
Sorry, that was a great question. So we do generally know how I'm going to speak mostly about sediment, not about hard rock, because hard rock has a lot more connectivity. But this sediment in particular, and the sediment we were looking at that spans this 3 million year span, that has almost no connectivity other than potentially some of the very top mud that's not lithified. So the lithification really prevents the microbes from moving around a lot. So it's basically whatever got deposited then is able to what persists. There are regions of sediment, especially in this site, where researchers notice that the sediment is so lithified that it actually cracks like rock, and so there might be some connectivity there as well. Yeah, that's a different kind of connectivity. Yeah, not very much, no movement.
All right, we have a question from online and then one more after me. What are your thoughts on how you would detect organisms that we don't currently have the technology to detect? How much do you think we're missing?
That's a great question. And that's one we're trying to really get at more, I guess the word is agnostic ways to detect life. And so I did not include this description, but when we look for this particular life, we're not sorting based on if we think it looks like a cell, we're sorting based on fluorescence, and specifically fluorescence of electron transfer. And so this is just what I think is the next step in kind of being more agnostic towards life we might be missing. And so electron transfer is, we're all doing electron transfer, and it's something that even a microbe that we don't know about or a microbe elsewhere would likely have to do. That's a great question. There's other ways we can look for morphology of cells. We can look for unexpected DNA or unexpected chemistry, like maybe different chemistry than what we have on Earth.
Yes, you talked about the slow rate of metabolism in anaerobic microorganisms. Microorganisms in aerobic environments tend to have a pretty fast rate of cell division, minutes, hours. Microorganisms in anaerobic, what is the order of magnitude for cell division, the timeline?
That's a great question. So I would consider myself a somewhat classically trained microbiologist. I grow a lot of microbes in culture, and I have grown aerobic and anaerobic microbes that have doubling times. Both of them can have really fast doubling times. So just an example, microbe in a hot spring, they're operating on much faster scales than a microbe from the subseafloor. And so they will double in a matter of hours, similar to like E. coli in the lab would grow. They just use different chemicals to do that. On the flip side, aerobic microorganisms can also grow pretty slowly, but generally, yeah, a lot of the examples of aerobic microbiology grow pretty fast, and a lot of examples of anaerobic microbiology grows really slow, but they can also kind of flip depending on how much sources of nutrients and chemicals you give them, according to their temperature. Yeah, you can manipulate all that. Yeah.
So this is the second of 4 talks. The next week, on next Wednesday, we're going to hear the new era of discovery, how technology is revealing the fragile animals of the deep. And this is John Burns. He just went, it is some of the most stunning videos I've ever seen of the midwater or the twilight zone. If you go below the lighted surface waters, it is this massive biome that we know so little about. And it's really amazing. So you have to come see that. And then our last one is resilient reefs, how algae grazing fish maintain balance in coral ecosystems. And so this is, many of you might have heard Doug Rasher give a cafe sci, I want to say now 4 years ago. And this is kind of the next generation of the research that he's doing. And it's going to be a great, great talk. And then one final point is that we've got these laboratory tours. So if you are interested in learning more about Bigelow, we've got tours coming up August 5th and August 12th. What's today? Okay. I'm pretty sure it's Wednesday. Anyways, you can sign up for these tours. And if these times don't work, and especially if these times don't work and you've got your kids or grandkids and you want to show them Bigelow, send me an email. You can find me on the website. We will get you in somehow. We don't want to miss an opportunity to get kids walking through these labs because they're amazing and that's the next generation and that's what we've got to encourage.